Hi, I'm Kenneth Sewell, Vice President for Research at Oklahoma State University, welcoming you to this month's edition of OSU Research on Tap. Each month, I sit down with an OSU scientist, artist, or scholar for an informal chat in front of a live audience at Stillwater's Iron Monk Brewery. This is a great way to get a glimpse into the fascinating research these talented individuals carry out at OSU. I hope you enjoy this edition and tune back in each month for a new one. Cheers. We'd like to welcome you here for this month's edition of OSU Research on Tap, where we're going to have a conversation with Dr. Tyrrell Conway, professor in our Department of Microbiology and Molecular Genetics. A little bit of a mouthful, but we're going to talk about
microbiology.
Microbiology is good enough. Well, we struggle with how to get a title for some of these scientific topics that will grab people, but also say something about what we're actually going to talk about. And so this month was E-Coli and Friends. That had me right there, like E. coli has friends. And something about adventures in the microbiome, and that was your suggestion. And then someone in my office says, wait a second, what's a microbiome? So we're going to come back around to all of that. But I would ask you to help me welcome Dr. Tyrrell Conway. So by the way, my goal in setting up these Research on Tap events has always been to, at some point, disturb the evening of the fire marshal, and we might get there today, so we'll see. But so apparently, E. coli has a lot of friends, many of whom are here today. But we'll start this out by asking you, why study E. coli? I have to say, when I first heard that was your topic, I thought, well, he's really interested in, studying what causes disease, et cetera. But maybe there's a bigger answer to that. Why study E. coli?
Well, E. coli is really cool. So you guys know E. coli as a model organism. You read about it in the newspaper. And model organism means that we use E. coli to figure out how life works. And that's my life's ambition, is to understand how cells work. So E. coli is the best model system for that. You may know E. coli as a biotechnology platform, which means it can be genetically engineered to make drugs. Your insulin would be made by E. coli in factories. And so that's all kind of cool.
But I think most of us associate E. coli with, like, bad stuff.
Well, there are good guys, and then there are bad guys.
So E. coli comes in more than one flavor?
Oh, yeah.
All right.
Thousands, maybe hundreds of thousands. We don't know yet.
So when you say understand how cells work, so you, as a microbiologist, I'm trying to break this all down. So what you study are these tiny organisms.
The lawyers tell me I study little squiggly things when they ask to depose me for 0157 outbreaks.
Okay, so you
little
study
squiggly things.
So when you so cells, for microbiologists, the cell is the organism.
Mm-hmm. Right.
So, but tell me more about why E. coli. How you got into E. coli?
Well, it is a platform for genetic engineering, and I was working in a biofuels laboratory as a postdoc. And by the way, I got my doctorate here at Oklahoma State University and went off to go Pokes. Went off to the University of Florida, and we convinced E. coli by putting the right genes into it to make ethanol, and that's used in the biofuels industry today. But that's not what I was really interested in.
We're going to come back to ethanol later, but you were really
cheers.
Interested. Yeah, cheers. Yeah. There's something about alcohol that cheers. We'll come back to that. But so in studying these tiny organisms, these cells, I sometimes hear you talk about, well, this one likes this, or it eats that. Mm-hmm. You talk about them almost in human terms.
Well, they're my friends.
Okay, well, there you go. So when I first heard this terminology, I thought, okay, well, these microbiologists, they're dumbing this down for us. Like when they talk to each other, they don't really talk about this one eats that or this one likes that.
Well, I tell my students that microbiology is easy. If you tell them it's hard, they're not going to like it. And you know, I have a therapy dog, and I'm on campus a lot, and I always say that she's a microbiology major, and they'll say, "Oh, she's a smart girl."
So tell me what it means for a single cell, for a microbe to eat something. When you say that, when you say it likes to eat this, what do you even mean?
Well, let's see if we can get it in perspective. If you took a pin, stuck it into a colony on a petri dish, you would pick up 1 million of those bacterial cells. Each one of those cells, in order to grow and divide in 1 hour, will consume 10 million glucose molecules, that's sugar, per second. 10 million molecules per second.
On the tip of a pin?
No, on one cell.
On one cell?
One
Each
cell.
one?
Each one consumes 10 million molecules of sugar per minute. So that's what they have to do to be successful. So when I say they eat, what they're doing is they're bringing sugar into the cell, making energy, and that allows them to grow longer and divide in the middle, and one cell becomes 2, 4, 8, 16, 32.
So when you say colonize, I mean, again, these are terms that we think of, you know, American history when we were little kids and all that. When you say colonize, what you're talking about this success, this growth taking hold.
Well, to me, colonization means that E. coli has colonized the intestine of an animal.
So it's become an inhabitant.
Right.
And
without reintroduction, E. coli, once established, colonized, it will have a sustained population indefinitely until something pushes it out. Could be another E. coli.
So maybe this whole intestinal thing is why you use these great euphemisms of eating and colonizing. And so forth. Yeah.
We have a review paper published years ago called "What's for Dinner?"
Now, sometimes in our career, so you've been doing this, I won't ask you how many years, but this has been your life's passion.
I started when I was 12.
All right. 12?
Yep, whatever.
Anyway, but we have things in our lives that kind of change the direction.
Mm-hmm.
I mean, for me, it was like neck injuries and not being all that great at college football says maybe you should be an academic. You know,
Yeah.
maybe NFL isn't for you.
How's a football player become
Yeah.
a vice president?
Yeah.
Right?
So for me, I had some life-changing experiences in there, but you had one somewhere along the way here. Maybe talk to us a little bit about that.
So I went off to Florida to figure out how to make biofuels, and what I fell in love with was metabolism, which is how bacteria eat. So they bring sugar into the cell, convert it into energy, and I was more interested in that than I was in making ethanol. Drinking ethanol is fine, but making it,
yeah,
that's, you know.
Leave that to Dave and Jared.
Yeah, and I asked them if they're growing any beer today, and they get the joke, but everybody else makes beer. But anyway, so I wanted to figure out how metabolism works, and so my students and I were characterizing metabolic pathways. That means cloning genes and sequencing and all of the things that microbiologists do. And I got a phone call from a guy at the University of Rhode Island. His name is Paul Cohen. And Paul said, I think that your favorite pathway is required for E. coli to colonize the intestine.
So the thing that you like to work on is actually essential. That's his.
I went right from petri dishes in a laboratory to mice in the animal facility. It's real world microbiology. It's what does E. coli have to eat in order to colonize the intestine? And it's important because every human in this room, every animal on this planet is colonized with E. coli.
And so
These are the good guys.
Yeah, the good guys. The good guys, E. coli, are in our intestines. But I mean, this is maybe hard for people to wrap their heads around as much as biology's been studied, but we're really getting down to these fine ones. We're kind of new at this in a way. So we really don't know like how or exactly what's allowed them to colonize the intestines.
Well, when we started this work was about 20 years ago. We knew that E. coli grew in every animal on the planet, even fish if the temperature of the water is warm enough. But we had no idea of what they grow on while they're colonizing. So you can't just go in there and expect to stay in there unless you can generate energy. All life requires energy to grow, and E. coli all bacteria in the intestine have to grow and divide in order to keep their population high. Because as you know, we are a flow-through system.
A flow-through system, I like
We
that.
are a flow-through system, and so
That's a euphemism, I think.
it is indeed. And every day, E. coli is growing and replenishing what's lost and on its way to the sewage treatment plant. So if they don't get energy, if they don't find food, they're going to die, or pass on through. So the question was open. What does E. coli grow on in the intestine? And so we figured out the answer.
And the answer is, as
The
Alex
answer
Trebek would
is
say.
sugars and amino acids,
Okay.
which is a lot like what we eat, except that we eat meat, protein, which is degraded to amino acids. We eat polysaccharides, plant fibers that are degraded to simple sugars, but that's what E. coli grows on, the simple stuff.
Okay. So this led to this thing you've called the restaurant hypothesis. And maybe you've had
Yes.
some other terminologies here, but talk to us about that.
We tried a lot of different scenarios. We were, this Paul Cohen and I, are just trying to explain at first to ourselves, how do bacteria go into the intestine and compete for nutrients? So,
It's not like E. coli is the only thing in there.
oh no. The average human has about 1,000 different species of bacteria. E. coli is just a minor member of, and this is the microbiome. A microbiome is a community of microorganisms,
Okay.
period.
Community, I mean, we have a community theater, we have a community. When you say community, what does that mean biologically?
When we say a community of microorganisms means a few or possibly hundreds of species living together in the same environment, ecosystem. And although nutrients are rich in those environments, the competition for those nutrients is stiff because the numbers are so high.
So if I come into a community, take it up to the human level, and there's nothing in the grocery stores that I prefer to eat, and there's maybe no job for me, I got to kind of move along, right? So if I'm going to live in that community, I've got to find something that can sustain me. I got to find resources. And so when you say community, you're talking about that balance of resources
Exactly.
for intake and energy production.
If you can't make a living, you don't get to stay in the community.
Yeah.
Now, we also likened it to the old boarding house analogy. So the boarding house is men would live there, someone would cook dinner, and there'd be a lot of different dishes on the dinner or on the table. And let's say I like Brussels sprouts best, and you're bigger than me, and maybe you like Brussels sprouts too. So the nights when you're sitting at the table, I'm not getting the Brussels sprouts.
I am bigger than you. I will stipulate that. Yeah.
I've got to switch to asparagus. Now, it doesn't mean there's not lots of Brussels sprouts and asparagus on the table. It just means that there's competition for those. And so sometimes I have to choose something else. And that's what we're trying to figure out. That's
But if all that's left for you are the things that you just can't tolerate, maybe your system can't tolerate,
then you lose.
then you lose. You don't make it at that boarding house.
That's right. So that's when we started calling it the restaurant hypothesis. You'll come in and there are things on a menu, so to speak. And in the same restaurant, there might be different menus at different tables. And we're still trying to figure out how that works exactly. There's a lot of competition in the community, and we don't know, we call it granularity in ecology, but the idea is how small are these communities? We think of it as one big intestine full of trillions of bacteria, but the bacteria don't appear to be treating it that way. They seem to be going to restaurants within that very large space.
So like a community, there might be multiple restaurants in the community, and they're picking the restaurants that are more suited to them. Gotcha.
I think you got it.
All right. Well, we're talking about this like it's all one big happy family, and we're joining hands and singing Kumbaya. But E. coli and the gut and microbiome is implicated in all kinds of diseases and health issues, and even things as diverse as Alzheimer's and ADHD. So help me understand the connection between this gut microbiome, this community, and these health issues.
We say that the microbiome is protective, meaning that when your microbiome is healthy, you're healthy. And that can take on a lot of different meanings. But like Crohn's disease, irritable bowel syndrome, these are dysbioses. That is, when the community breaks down, these are problems with the microbiota. They're providing energy for the cells that line our intestine, the bacteria are. They're providing vitamins. So there's a lot going on in there that does contribute to our health. But sometimes things get out of whack. And you hear a lot about probiotics in the news as a way to restore that microbiota.
So this little pill in my microbiome is all, my gut's all back to health, right?
That's right. And they have great names.
And great dollar signs beside the price tags.
That's right. And every day you take another capsule, and the reason is because they never colonize. The probiotics pass right through you. It's just the perfect drug. You need it, and you have to take it every day.
Because it's not there the next day. When
That's right. But you need it because somebody on TV told you you need it.
So when does the bad guy E. coli come into the scene here? How does that work?
Well, let's say we go out to dinner later, and we get a hold of some contaminated hamburger, infected or contaminated with the 0157H7, which you all know what that is, I imagine, because those are the bad guys in the
That's
news.
an everyday term, right?
It is.
No, tell us. No, okay, come on.
You don't know that?
Yeah. 0157H7.
It has another name, 0157H7. So when you hear about an E. coli outbreak, that's one of many different culprits, but that's the primary culprit. So 0157 is not good. It has another name, enterohemorrhagic E. coli.
I'm good with 0157.
Okay. I can explain what that means.
Please do.
Bloody diarrhea.
That's easier for me to remember than either one of those other things.
And you would know if you had that. And you probably wouldn't be sitting here,
I think you're right. That's right.
So we're healthy and we go out to dinner and we share contaminated hamburger, and you don't get sick, but I do. Why is that? All outbreaks have an attack rate, and some 20% of people who are exposed to 0157 will become sickened and others will not. Most won't. We think that the microbiota is protective. So in the case of you're resistant to this infection, it means you've got E. coli that prevent the bad guys from taking over, but I'm susceptible.
So they're the big guys at the boarding house. So they're eating the stuff that...
You got more big guys.
Yeah, I got more big guys than you. Yeah.
I got these little puny guys who can't defend my gut. And
So the susceptibility is it an accident? I mean, is it truly, does it mean that something's wrong with your diet? Or is it truly just a random thing where some of us, the community has developed in such a way that it's protective of that particular strain and others are not? Or is it an actual problem in and of itself?
we should know the answer to that question, and we don't.
Well, but that's why you get to come
That's
to
why
work
we do
every day, right?
research, yeah. NIH wants to know, so they fund our research to figure it out. So we're working on the basic principles, but we hope that someday in the future we could analyze your contents, your intestinal contents and mine, figure out what bacteria, I always say who's there, but what we mean is what species of bacteria are part of our communities to interrogate whether or not we're susceptible or resistant. And when you think about outbreaks of hospital-acquired infections, that might be really nice to know if you've got an outbreak on a hospital wing, to ask of all of the patients on that wing, well, do we keep them here because they're resistant, or do we move them someplace else in quarantine?
Now, I always heard that lawyers are trained to never ask a question they don't already know the answer to, but I'm not a lawyer, so I'm going to deviate from that. So in this fantastical future that you're laying out where we know enough that we can analyze the contents of someone's microbiome, and how would you do that?
Right now, you would just sequence it.
But how would you find out what's in my microbiome today?
We would just take a fecal sample.
Oh, that sounds like a euphemism as well.
I can't get my wife to do it at home either. There are these things like GoFundMe projects, and one of them was the belly button project, and people were swabbing their belly buttons and sequencing the bacteria that are in their belly buttons.
That was a GoFundMe
And millions of
project?
people
Donated money
participated.
to that.
It's like a public research project. There are public microbiome projects, and these are intestinal microbiome projects. And if you buy into this, you're going to get a box that has a little cup, and you know what to do with the cup, and you send it off, and they sequence it, which is just puts a DNA sequence tag on what the organisms are that are there. So it's a way of naming them.
Okay. So we're doing good on time. I'm going to ask you one more question. We'll take a little tiny mini break here. But give me the success version of this. Not that it's not been successful. You've learned a lot. You've made some breakthroughs. But where could this be? And you've been working on it over 20 years. Where could this be in 20 years?
I would like to know why it is that some individuals are susceptible to enteric infections. What I mean is diarrhea.
Okay.
There are so many different forms of diarrhea, and why others are resistant. If we could go to Africa and change the microbiota of children to make them resistant, we could save 2 million lives of infants a year.
Yeah, because in this country, for the most part, we talk about diarrhea as an annoyance.
It is.
And-
You go to the hospital, we know how to treat the symptoms, not necessarily the disease.
But we don't lose a lot of people. But
We'll give you an IV and we'll keep you alive.
at a worldwide level, this is truly life and death.
Mm-hmm.
Fantastic.
So that's the big reason.
Cool. I like that.
Coming up next on Research on Tap.
Because I went to the Stillwater roller derby match on Saturday. And you said there's actually a study on the micro...
The
Yes,
exchange of
exchange.
microbiota between roller derby players.
Okay, this blows my mind. You mentioned the ethanol work. I heard some rumor about you being a part of the 5 millionth patent in the United States. Is that part of your E. coli work, or is that part of something else?
That was genetic engineering to make ethanol. And
Genetic
the Patent and Trademark Office chose that patent to celebrate their 200th birthday.
So you are a part of the 200th birthday of the Patent and Trademark Office and the 5 millionth patent in the United States.
With all the zeros, just like the 5 millionth hamburger at McDonald's, I suppose.
And I'm sure they gave you $1 for every one of those numbers, right?
Oh, they told us we were going to be rich. And we are, but it's not money.
Well, before we transition to Q&A, I can't leave it alone. You've talked about ethanol. We've had our little reference to our beer here but as part of your microbiology work, you also have a little connectedness to the Iron Monk Brewery here, right? If people don't know about that, you should absolutely tell them one of your teaching passions.
I've been teaching a course called Brewing Microbiology, and it was made possible because Dave Monks and Jared Milirons opened this tap room. And if you can grow E. coli in the laboratory, you can grow yeast in the laboratory. So this is a course about the science behind brewing. There are hundreds of thousands of courses out there where you can learn to brew beer. This isn't about that. This is the science behind it. And we do give a tour, video tour. This is an entirely online course. We have somewhere around 150 to 170 students per semester takes this course. It's also offered as a massive open online course.
Or a MOOC, they call
and you
it,
can
yeah.
take it for free.
That's a pretty good connection to the Iron Monk Brewery for our guest tonight. So, all right, I think it's time to open it up. So I've gotten to ask the questions that I want to ask. We have, where's the ball? We have a microphone ball right here. Amy's got the microphone ball. We are recording this for eventual broadcast. In a few days, we'll have this up on O State TV. So we do want our question askers to have the ball in hand as they ask a question. That way we have the audio. The ball, if you talk with your hands a lot and do this, it will quit working because it thinks it's being thrown rather than held. So if you get the ball, hold it still and ask your question. And who would like to ask a question? Right here in front.
All of us in this room have taken antibiotics, and antibiotics are to kill microorganisms. What can you tell us about taking antibiotics and the microbiome, how it recovers, information in that general area?
There is absolutely no question that when you take oral antibiotics, you will impact your microbiota. Sometimes that's harmless, but in many cases that opens a door to disease like Clostridium difficile. C. diff is not an infection to be messed with. The recovery time, at least in animal studies, is somewhere between 3 to 9 months. So I quite frankly avoid taking antibiotics if I can.
So there's some kind of vulnerability that's at least theoretically opened up by taking antibiotics.
There is. Now that doesn't mean that you shouldn't take antibiotics when you need them. Let's say it's better than, dying, but.
What's the recovery time?
It's 3 to 9 months from the end of the course of treatment. The measurements on, so again, these sequencing studies, we say, here's what was in this individual. Now we're treating with antibiotics. There is a definite decline in the makeup of the microbiota, and what that really means, of 1,000 species, hundreds might disappear. And it takes up to 9 months to get that healthy microbiota back.
To get the same kind of profile of, that's it.
Are you aware of any probiotic supplement that will protect you from an OH157 infection?
There is a culture that's sold in Germany. It's called Nissel 1917. And it was isolated during World War I from a German soldier who survived a dysentery outbreak. And so there were some great microbiologists in Germany who actually removed this organism from this guy. Well, it was isolated from his feces. It is still sold today under the trademark of Mutaflor.
Beautiful.
And the idea is to offset some of the ill effects of traveler's diarrhea. And it seems to work. There are also some indications that some patients are immunocompromised and they have died. So it's not the safest. It is temporary. But I should add that we're all colonized, everyone in this room, on average, with about 5 good E. coli. But if we come back and test you in 6 months, it might be 2 of the same and 3 different. So there's a steady succession of E. coli strains. And there are literally thousands of E. coli strains. But that's normal. That's part of the normal turnover of your intestinal contents and the makeup of your microbiota. So this would be in a case where there is a dysbiosis being caused by an infection, and the idea is to try to get this good guy to come in and chase the bad guys out, so to speak.
Okay, it's my job in this whole thing to make sure when you use a term like dysbiosis, we know what that means. I think by context, we got it, but go ahead and define dysbiosis here real quick for us.
That's when the healthy community structure of your microbiome is disrupted.
That's what I thought, but I want to make sure.
A few months ago, I went to a science cafe that dealt with prebiotics and talked about making a better microbiome using that. Could you comment?
So in theory, probiotics and prebiotics are a great idea, okay?
So tell me what a prebiotic is.
A probiotic is a microorganism that you ingest with the idea of it making you healthier. A prebiotic, she's been reading, I guess, is perhaps a dietary fiber that you would consume to try to promote the growth of an already resident microorganism.
So adding items to the menu at the restaurant.
Exactly.
All right.
So the existing theory, the one I believe in, is that the reason you can get 1,000 different species is that each one grows on a different nutrient better than all of the others. So if you want to promote the population of a healthy organism, you should feed it. And I know those experiments work when we do this in mouse studies. So we can feed particular sugars to favor the population of particular E. coli.
So it's at least a concept that has merit. Whether the things that are on the market now are successfully creating that, do you know of any evidence to suggest that?
I'm not convinced that anything out there is working well right now.
Okay. But there's some merit to the concept.
The theory is awesome.
Back here.
Yeah, one more question. Something you just said, there's not just one collection or one spread of these microorganisms. There are various combinations that will be protective and healthy for you. Is that correct?
That's right. So I might have said earlier that there are about 1,000 species in the average human intestine. That's in the Western world with a Western diet. If you go to some places in the world where food is not as easy to come by and some pretty strange stuff on the table, typically those people are going to have about 3,000 different species of bacteria in their intestine. If you look worldwide and you just tried to name all of them, it's about 20,000. So you have a subset, 1,000 out of the possible 20,000 different species of bacteria that can possibly colonize the human intestine.
And that depends on what we're eating.
So it looks. So that's still controversial, but I don't think there's any question that as the evidence accumulates, we're going to say, yes, your diet affects.
At least to some extent, it depends upon what you're eating.
There's so much else that goes into it. It's the people you surround yourself with are influencing your microbiota. Your genetics are influencing.
I can't help it. I got to bring this up because I went to the Stillwater roller derby match on Saturday, and you said there's actually a study on the micro.
The exchange of microbiota between roller derby players.
Okay, this blows my mind.
And rather than numbering the subjects in these studies, they just use their roller derby names.
Oh my God. So I just have one question. Was Pitbull one of the names?
I wish I could remember. Okay.
But, so it's not just what we eat, but it's also who we're interacting with and the physical environment as well as the nutrient environment.
And your genetics.
And genetics.
Yeah.
Where's the ball? The ball's over here.
So, nutritional sciences major here. Have to ask the food questions to piggyback a little bit on what's already been asked. So, have you studied more so, you've talked about what E. coli eats and those sorts of things, but what are the outputs of E. coli in the sense of how does that affect the health of the gut and the overall individual, and how does that play into inflammation, oxidative stress, all of these things that we've done a lot of research on and we look at very thoroughly. So I'm curious about what you've seen in your labs.
Well, that's a pretty complicated question.
So I'm going to boil that down. So E. coli eats things. It also eliminates things.
It excretes things. Yes, it does. Yeah. And some of those things that E. coli makes as byproducts of metabolism will feed other bacteria. It's the anaerobes that are making butyrate, you know what that is, I'm sure, and that's feeding colonic cells. So when I say...
I give up on the whole technical term
I know.
thing.
What that means is that the bacteria that don't like air, anaerobes, which are the most abundant organisms in our intestine, they're fermenting undigested fibers mucosal secretions, that kind of thing. So complex polysaccharides, they make butyric acid, which is a little thing that they make as an end product of their metabolism. That's what our intestinal cells eat.
The cells of our intestines, in order to maintain integrity within the cell
The
structure.
bacteria are feeding our cells, and that's about 10% of our energy balance.
Wow.
It's kind
I'm
of
sorry,
cool
I
to-
didn't mean to interrupt you from her question, but that's fascinating.
So I don't really know the connection, and nor does anyone. So cellular immunology, innate immunity in the intestine is a wide open field, and it's not my field. I just hear these guys talk at meetings, and I hear them say enough to understand that they don't understand it either. So there's a lot to be learned about how it is that our microbiota could become dysbiotic, that is, to create inflammation. But we know inflammation in the intestine's bad. But the connection's not that clearly established. Now, the fact that these patients are recovering upon antibiotic retreatment tells you that there's definitely a connection, but how that is restored, that's not understood.
So I think he just gave you a career's worth of research questions to work with. Over here.
All right, I want to go back to your prebiotics. So, and you apparently like prebiotics from what you said.
At least the concept of them.
The concept of it.
Any fried food, I think, is a good prebiotic.
I like it. So if you have this community of bacteria, even in a normal, healthy person, some of those bacteria are not good bacteria,
That's
just in general.
almost certainly true, yes.
Okay. So if you are putting in these prebiotics, what is the incidence that what you are doing is selecting for the bad ones?
Well, that
You know,
would be
providing
bad.
them the food. Yeah, that would be bad. So,
So how do you
often does
feed
that
the good
happen?
ones without feeding the bad ones, right?
Yeah.
Well, that's the goal, right? But, you know, antibiotics are supposed to kill the bad guys without harming the host and killing the good guys. And there's always some collateral damage. But I don't know the answer to the question.
Okay.
I don't think anyone does. Theoretically, we could target one organism with one prebiotic, but that doesn't mean that there aren't many other species in this community of organisms that could also eat that.
So Paul Cohen's question, idea that you guys have been working on all these years, really does fit in with the notion of, so like, fried chicken might be my favorite food, but I'll still eat, hamburgers or McNuggets at McDonald's, even though they're not really fried chicken. Yeah. So that's the idea, right? So to have that specific a target, the microbes don't have that specific an appetite. There's something they like best,
Right.
but not something they only can live on.
Right. It's a smorgasbord or a buffet table,
So we've got restaurant, smorgasbord, buffet table, boarding house. All right, let's keep it going.
So what's for dinner?
I wondered if you could comment on the role of consuming fermented foods.
We talked about this a bit, didn't we? Yeah.
It's not something I've ever done nor studied, to be honest.
You've never had anything fermented, all right?
Well, the guys in the back make this, I can't remember, but it starts with a K. It's and they make it with different flavors and stuff, and it's not for me. I'm sorry, I can't comment on it, but it's...
Okay, back here,
Hi, Tyrrell.
Where are you? Wave your hand.
back here.
Okay.
So can you summarize real quickly the contribution of bacteriocins between different bacteria and how they establish this community and maintain it?
Yeah, okay. So bacteriocins is the new subject, and that's even easier to understand than dysbiosis. So Peter's referring to something that one bacterium makes to kill another bacterium.
Ooh.
Biological warfare. And so the question is biological warfare a way to establish a microbiota? And the answer almost certainly is no. And the reason is because it's really difficult to understand how to poise the concentration of a toxin to achieve a steady population, because this follows an S-curve, which is an order of magnitude, the response of that population to a bacteriocin. Too much, they're gone. Too little, and there's no effect. So to poise the population with an inhibitor theoretically doesn't work, and to me it doesn't make sense. So I hope I answered your question.
In the back.
So if I take this course of antibiotics, why can't I just take a pill to introduce the good organisms back into my system?
For the same reason that pathogens have an attack rate, probiotics don't colonize normally healthy hosts because there's a barrier to colonization. That same barrier to colonization by probiotics works to protect most people from infection.
So there's something in there already eating the food that those probiotics would want to live on.
Probably. But there's no way to answer your question for any individual. So I would say if you think that yogurt makes you feel better, eat it. I think if you're taking antibiotics, that eating yogurt's really not a bad idea. So those dairy organisms can colonize under the right circumstances. What I suspect is over time, those dairy organisms are displaced by what were there originally, which are more compatible with the human intestine than cow organisms.
Well, I don't know how it's possible to feel like I've learned a lot and to feel like I'm really stupid all at the same time. But I feel like that's happened tonight. So somebody else has a question. I'm not cutting things off just yet. Go ahead.
Okay, you talked about how probiotics can't colonize well enough. What about like with C. difficile, and you treat that with fecal matter transplants? What's the difference between that successfully working and like a probiotic?
So we have to talk about fecal transplantation. Now,
somebody had to be the straight person to get you into fecal transplantation today.
So I can tell you that I go to meetings where we sit around, we eat lunch, and we talk about this stuff. And
stuff. I like that. Yeah, good.
So I can tell you that there are some well-documented studies where a patient with a C. diff infection has been treated with essentially a fecal enema from a healthy donor. Typically, this would be a spouse or a sibling or someone in the same house.
So they're going to have a similar microbiota to begin with.
When you're young, it's your mom. When you're an adult, it's your spouse.
Yeah.
Or,
Yeah.
If you're a college student, it's your roommates possibly, depending on how frequently you go home. That's why we don't do these studies at home. There's a yuck factor that goes with it. But those studies are really solid, and the measurements taken during C. diff infection show a typically reduced overall population, so community structure is diminished. So there's many fewer organisms, and it's restored to a more healthy microbiota after the fecal transplantation. So that really can work. And getting back to what Ron was saying, that doesn't mean C. diff is gone. It just means that the microbiota has been restored to a more healthy balance. However, things get off again, and C. diff is still hiding. When I say hiding, we're talking about 10 to the 11 bacteria per gram of feces. That's a very big number.
But in a small enough number, relatively speaking, to be in the background.
So not enough to cause damage to the host, disease, and yet enough to remain hidden and ready to bloom again if circumstances allow.
Well, I have to say, we do a little preparatory interview before we do these sessions, and I thought we might make it through this without a conversation about fecal transplantation, but we didn't make it. Help me thank Dr. Tyrrell Conway for his talk tonight. So I mentioned that our next Research on Tap will be here on March the 12th. We'll be right here in the Iron Monk Tap Room. We'll be talking with Dr. Danny Belmer, who is a professor in biosystems engineering and biosystems engineering, and she's also in the Robert M. Kerr Food and Agricultural Products Center, or FAPSI as they call it, and she will tell us about spirulina. Spirulina. So the rest of the title is that an Italian term for spiral macaroni? Or a bacterium that could solve world hunger?
I choose B.
You're going to go with B. You don't want to miss that. All right. So before we finish up, we have a traditional gift for our speaker this evening, which is the Research on Tap medallion that is also has a, if you hold it just right, there's a bottle opener on there. Okay. So we will see you back here in March, but until then, our traditional toast, may your intellect be meritorious and your impact be broad.